If you are in Meadville, it's a beautiful sunny day. Not. It's kind of funny. I was thinking that yesterday and the day before we had this beautiful sunny day, and now it's like kind of gray and, well, just not exactly a user-friendly type of day. But I think it's still going to be fun in this class because I think we've got a really good, we have some really good discussions to be had today. And so I invite you to go ahead and to pull over your slides in the class docs repository. They should all be there, and we will get started. Today what we're talking about is something that I find actually quite exciting. When I first saw this particular topic, I was just absolutely, I was absolutely floored. What we're talking about today is something called horizontal gene transfer. Now, this is relevant to us because let's look at the what we've been talking about so far in this class. We have been talking in this class about how one sequence of genes can become different from another sequence of genes. And so what that really means is that some kind of mutation has happened. Up until now, we've been talking about this as being a mutation has happened to one sequence. And that sequence then became different because of these random mutations. But we'll see today, though, that that is not the only way that you can have one sequence become different from another sequence, which is just extraordinary. And it's called horizontal gene transfer. And the idea is that you get this, the genes that move from one organism to another, like in its entirety. But before we get to that, I wanted to make this announcement. Where the first 3 slides are about the exam. I'm thinking that the exam will be given on Monday, the 12th of April, which is it's during the lab, but not the next week's lab, but the week following that, the lab following that lab. And the reason why I say it's not next week's lab is because we have a speaker who's going to be coming in to talk about her research. This is Yimon. And so it's going to be a week after her talk. And that is going to be our exam 1. So it's basically the differences between RNA and DNA. You'll have some basic Python programming syntax, keywords, and definitions. I'm thinking that you will not have to write any code in class. You just have to recognize that the code or what the code will do. And perhaps even whether it has the right kind of syntax, but this is not necessarily this is not programming 101. So I don't expect you to have to actually write the code or to do some amazing bug defining here. But I'm thinking that what I'll do is I'll give you some code, and then you just have to go through and figure out what it does. You just have to follow through. But you're not supposed to use your computer for this. You're supposed to use your brain. So it's like that. We'll also have global and local alignment. We'll have terms and definitions. So really you're just kind of like comparing definitions and terms and connecting them together. So it's multiple choice. Or multiple choice in that way. Oh, not multiple choice. Well, it could be multiple choice, but it could be like actually matching. We'll have some questions which are similar to those that you've seen already on the activities. You'll have some questions which concern the central dogma of biology. And by that I mean like transcription, translation, those 2 big pillars that we've been discussing in biology, where transcription is where you're taking DNA and you're really converting it into a different type of written form, which is called RNA. And then translation, when RNA becomes a completely different language, which is called protein. And so that's the big differences here. And so you'll have questions which concern these kinds of issues. I'm not going to ask you, by the way, to actually take a piece of RNA and convert it into protein. Because no one has time to memorize that table. That's why there is a table of amino acids. And so I think that you can think of these questions as more theoretical types of questions. Then you have detecting genetic disorders. We've done some experiments in class where we've actually been detecting genetic disorders through our code. But basically what I'm trying to say, though, is that study your slides. The slides are the scaffolding, and the book has the kind of the extra, the detail. But follow your slides, and then the slides will provide you with the knowledge that you need to do well on this test. So what the test is going to be looking like when you take it, it's going to be a multiple choice. It's a short answer and short essay. I guess I should also put like matching in there too for the definitions. But it's going to be given on Sakai. Sakai has tests and quizzes. It's one of the tabs that you can find in Sakai if you go to Sakai to check your grades using Postum. You'll see that on that same side of the screen there's also tests and quizzes. Now this is kind of a weird thing, but you're going to have 24 hours to take the test here. Now, why would you want 24 hours? Is it going to take me 24 hours of agony to get this thing through? No, it will not take me 24 hours. In fact, it's probably going to take you about 2 hours to actually get this whole thing done beginning to end. But the reason why I'm giving you so much time is because we have people in this course, your colleagues in this course are from maybe, I think, 4 different time zones. And so it's fair for them to be able to find a time during this 24-hour window where they can take this test in some comfortable and peaceful environment where they can actually concentrate. And so it's not like a timed test, but once you actually click the test-taking button on Sakai, then the clock will start running, and that's going to be about, I don't know, 3 hours for it. I don't think it really matters how much time it takes you. I don't think I've set a setting that says, stop the test after this much time. But what I'm saying though is find yourself a good 2 or even 2.5 hours. You're probably not going to need it, but find that kind of time all together in the day so that you can take this test during that 24-hour time somewhere. And I'm hoping that that will be a convenient solution for everyone in this course so that they can find that peaceful place to do it. Yes, sir?
So just to clarify, do you want us to finish it under 3 hours, like all in one sitting?
Yes, and so I would like you to, you can have as much time as you want to take it, but I'm saying that how much time, so you will, I'm saying that when you take this test though, you will probably find that you spend under 3 hours to take it.
Okay.
But if you need more time, you can have it. It doesn't have to be done in 3 hours. You could take 5 hours, but I wouldn't recommend taking 5 hours for any test. That's just going to go. That's just.
Is it open notes too, or is it not?
Well, it's going to be theoretical, so it's going to be taken at home. So is it open notes? You could use your notes. You can use your notes. Yeah, because you're going to be in your dorm room. But I'm going to be writing this test in a way that maybe you'd have to spend some more time, thinking about the answer before you won't be able to copy and paste the answers out of the thing though. So it's going to be, some of the questions are going to be theoretical. So it's going to be, it could be open notes if you want. But I think that it should be an interesting test to take, and you have as much time as you want to take it, but in that 24-hour time. So that means it's going to be coming out during lab, like this, at 2:50 on Monday, and then on Tuesday at 2:50 it's closed, and so you have that time. And I'm hoping that for all of the colleagues in this course who are in other time zones, I'm hoping that that's a convenient way for you to take this exam. But if it isn't, then please let me know, and we can find other solutions that might be better. Okay? Yeah. So please let me know. Please let me know. I don't want this to be. This is not a weeder course. It is my front and foremost interest that everyone in this course do well. I want you to do well. I want you to enjoy this. I want you to move forward and realize that there's a career waiting for you in bioinformatics if you want it. And so I don't write tests that try and weed people out, but rather I like to think that the tests are more there to help you to showcase your exceptional knowledge and show how you can use the skills that you've gained in this class. Anyway, but still, if you have any questions, please let me know. I'm available. Okay. All right. So what I want to talk about today, though, is horizontal gene transfer. And so, like I was saying, horizontal gene transfer is actually quite interesting because up until now, we've been saying that the only way that one sequence can diverge from another, that in other words, a sequence can become 2 different sequences, is by mutations. That means you have the original sequence and you have another sequence that has mutations, and then they become 2 separate sequences. And while that's true, there's another way, and that's called horizontal gene transfer, which is really quite remarkable. And horizontal gene transfer is basically, it's a transfer of gene content, seemingly in its entirety, from one organism to another. It's like you're transferring knowledge, for instance, in this class to each other, from you to me. We're telling each other things, and we're learning things from each other. And so that's a transfer of information. But imagine now we're transferring genetic content between organisms. That's horizontal gene transfer. And so when I say the word horizontal, what that means is like you have 2 organisms which are not related at all. There's no relation really. Of course, everything is related in biology, but there's no recent relationship. It's just 2 separate organisms. And the genes come from one organism and they travel across this tree of life to the other organism, which happens to be in close proximity, and they get those genes. And so that's what they mean by horizontal. Vertical gene transfer is kind of when the genes are passed down from a generation to another generation. It's up and down. It's like you're thinking about your parents have the gene, and then you have the gene. And so it's like it's handed down to you. But horizontal is where two unrelated organisms suddenly are displaying the same genes, and you're thinking, you're not related to each other. How are you resembling each other like this? And so what we're talking about today, though, is basically this antibody, like the antibiotic resistance. This actually happens very commonly in bacteria. In fact, horizontal gene transfer happens so readily in bacteria. One of my projects I was doing during grad school was to try and figure out what commonalities existed between a Staphylococcus genome and an E. coli genome. That was the one question that I had to answer for one of my projects. What common protein existed? And I started to find that in E. coli, it's like everything that was in the other bacterium had been copied, and basically it was sitting in E. coli. It's like E. coli, it was like this giant vacuum that had taken in all the protein stuff, all the different types of genes that I was looking at. It was all in there. And I thought, how can that be? I mean, staph, or whatever, Staphylococcus and staph and some other things I was looking at, were not actually related to E. coli. These are two separate organisms. E. coli is the bacteria that lives in your gut, whereas these other ones these other bacteria I was looking at may not necessarily live in your gut. But the thing is, though, that they have spent some time together, and when they spend time together, E. coli grabs whatever it can from seemingly all these other organisms, and suddenly E. coli has the same genes that Staphylococcus would have, or some of these other organisms. But what's actually going on, though, is that DNA is going from what's called a donor to a recipient, where you have a chunk of code from a donor cell, which is in this case bacteria, and that bacteria gives some DNA to the recipient. And then the recipient starts demonstrating the same types of traits that the donor had. So horizontal gene transfer, it's the transmission of portions of genomic DNA between distant organisms. They're not the same. They're not related. They're distant. But yet now, when you somehow couple them together in a certain way, when these organisms are together, and by this I'm talking about the organisms I'm talking about are bacterial. But when these organisms are coupled together in a certain way, they start sharing their gene material, their genetic content. So this is where I was talking about the vertical inheritance, where you're talking about vertical inheritance involves mating, whereas horizontal does not. But really, just remember, though, that various fragments of this genome are being moved from one organism to another, and each organism has a different evolutionary history, but somehow they start showing that they are the same. But basically, the most important thing here is that bacteria can acquire new traits such as antibiotic resistance or pathogenic toxins. That means that they somehow get some special advantage from their neighbor, in that they can still thrive in spite of antibiotic medications being delivered to the body. So in other words, you can still have an infection from some kind of organism in spite of the fact that you're taking meds or antibiotics. And so this is where you have the notion of superbugs, bugs which are resistant to penicillin or ampicillin or any of those things. But superbugs come about because one of the organisms that they were near happened to have a mutation somehow which allowed them to survive penicillin. But then they got that gene, and now they can all survive penicillin. And so it's for that reason that penicillin doesn't work as well as it used to, and you have to in fact have a prescription to get penicillin, because if it were sold over the counter, then people would take penicillin for seemingly anything, and they might get it wrong, and they might take, they might say that I think you have to take like maybe 2 weeks of penicillin to kill the infection, and you start feeling better after the first week. But that means you haven't killed the infection. You've just made those bacteria weaker, and so then you have to finish your prescription. Because if you don't, then the weaker ones will grow stronger, and those ones will be the ones that will be resistant to the next dosage of penicillin. And so doctors give you prescriptions to make sure that you're taking it correctly, so that we don't generate more superbugs. Anyway, but that's actually quite cool. And that's all as a result of this horizontal gene transfer. And so this is this finding, we're looking at fragments of genes that do not belong in a genome. And there's a couple of ways of doing this. There's something called parametric, a couple of ways of finding that the horizontal gene transfer actually has occurred. And that is that we can use what's called parametric methods, where basically you're just going through the genome and you're looking to see whether you expect to find the kinds of combinations of A's, T's, G's, and C's that you're coming across. And so this one here for genome location in the upper left-hand side, this is your GC content. We've discussed already that GC content, G's and C's have 3 bonds, which are stronger than the A's and T's, which have 2 bonds. And in some sequences of DNA, you don't expect to have so many triple bond GCs, or so many triple bonds in the content. That means you don't expect to have so much GC content in the DNA. You might find that there's one organism that has a whole bunch of GC content, just naturally it does, and have another organism that tends to favor a different kind of, like maybe A's and T's, but seldom CGs. But you find that when you have horizontal gene transfer, that you have these chunks of code which just kind of look out of place. It's just like, where did you get all that GC content? You'd ask the organism. But you'd find that through these statistical methods where you're just kind of creeping along the sequence and you find the G's and C's are there. There's another thing you can look at. For instance, you can use phylogenetic methods. The word phylogenetic is really about looking at trees. It's about creating a family tree where you're looking to see how things are related to each other. And you create this family tree of all these different organisms. And if you look at 2A, for instance, you'll find that there's what's called the species distance, and that is that if you analyze all the sequences from all of these different organisms, you'll find that they all have a certain kind of natural distance from each other. I mean, after all, they're beginning to evolve separately from each other. And so the sequences become more and more distant, perhaps as evolutionary time goes along. But then you find that there's 2 organisms which are not supposed to be the same. They're not supposed to be related because they are evolutionarily different, yet they have common genes that are almost exactly the same. And so in this species distance here, you find that those 2 organisms that has HGT, or horizontal gene transfer, those guys are right next to each other in terms of similarity. And you're thinking, how could that be? They're not the same organism, but they have the same genetics. And so then there's another one here where you're looking at phylogenetic models and you're looking at trees, and then you just basically run a full analysis on all of the sequence data, and you find out that there's 2 organisms which are actually quite close to each other in terms of relatedness, but they have no real business being together. So a famous experiment was done, and you may have seen this before. This is one of those poster child type experiments that every biology textbook would have on, say, chapter 3. But basically, though, you have 2 different strains of organism. And you have strain 1 and strain 2. Strain 1 has these genes bio and met, and strain 2 has Fe and Thra. And the thing is that these genes you need these genes to survive, but strain 1 needs the other 2 genes. That means it needs Fe and Thra to also survive, but strain 2 needs bio and met to survive. So in other words, if you are a strain, you need to have all 4 of those genes present to survive. And so here are 2 strains of those organisms for which half of the genes are present. And so we have basically strain 1 and strain 2, which have, if they could somehow pool their resources, then you'd get an organism that would survive. And in this experiment, you actually do get that. And so what you're seeing here is there's a colony of organisms which appears, which has borrowed, which is basically from, maybe it's strain 1, which has borrowed 2 genes from strain 2, or maybe it's strain 2, which has borrowed 2 genes from strain 1. But one of these strains has borrowed the missing genes that it didn't have from the other type of gene, or from the other organism, and suddenly you start getting these bacterial colonies growing on these plates. Otherwise, growing strain 1 and strain 2 by themselves, they don't grow because they don't have the other 2 genes. So it's like you're building your genetics. You're building up your genetics in some way, and if you don't have all 4, then you can't grow. And so you're actually able to grow out you're actually able to grow when you borrow these genes from your neighbors. This is interesting. This is another experiment that follows the first experiment, and that is that they were wondering, well, how does this horizontal gene transfer happen? And this is saying that you actually have to have organisms in contact with each other. This is an experiment where you have a rounded tube, and on one side you have maybe the organism that had missing bio and missing met genes, but it had the Thre and the Thrust genes. Whereas the other one, you have the bio and the met, but you don't have the Fe and the Thrust genes. And so, because you have the separator there, the genes cannot mingle, or the organisms cannot actually physically touch each other. But when you remove that filter, when the organisms can actually float around and work with each other, then they're able to share their genetics. And so this means that you have to have they have to be in physical contact with each other. And so in order for you to have these organisms growing in these bacterial colonies, you have to have physical contact, and that's kind of the whole area of research here in horizontal gene transfers, and that you're trying to think, okay, well, here are these organisms that are sharing common genes. When were they ever together? When did they ever meet? In order for them to undergo what's called this conjugation. And conjugation, I'll just read this, is the process by which one bacterium can transfer its genetic content to another. And it's through this, what they call direct contact. There's another way of sending information through, like genetic information, and that's transduction. We'll talk about those. Transduction is when a virus basically infects one organism, and then it somehow takes some of its genetics with it. The virus kills the organ or kills that cell, and then the cell disintegrates. The virus is floating around. It infects another, or those viruses infect another cell, and then when it's infecting another cell, the genetic content that the virus has picked up is implanted into the new host, and then that's how genes would move around. Either way, it's like the content, the genetic content is exactly the same, but it just moved by a virus. And then we have transformation, where it's like it's where DNA is really just kind of floating around from some kind of an accident that happened where the cell died and it just kind of disintegrated, or maybe it was lysed or something. But the DNA came out and it was floating around and somehow managed to make its way into a new cell. And so we have these kinds of things here. So we have this transformation where the cell explodes, so the DNA from the cell comes out in some way and it makes its way into a new cell. Conjugation is when the actual cells, we're going to spend a lot of time talking about conjugation, but when the actual cells transfer information across with some physical bridge. And then we have this transduction where a virus actually is the vehicle that moves the stuff. What we're talking about is like this conjugation, though, where you have this donor cell in the middle. You see this donor cell, and you have a recipient cell, and the donor cell has this red ring that's next to it. That red ring is called the F factor, which is really extraordinary. The F factor it's like this fertility factor. It's this catalyst that if the organism has that, if the cell has that, then it's able to plant that its genetics into another organism. It's just one of those things. And it also, when you have this conjugation, I think the F factor also moves across so that new organism now is able to, I mean, I don't want to use the word infect, but that's really what's happening. It's planting its genetic content in other organisms. So a very important book or paper came out, Horizontal Gene Transfer Among Bacteria and Its Role in Biological Evolution. This was a very important paper. You can look it up and read it. It's actually quite interesting. But it's basically saying that bacteria was kind of faking it till it made it. It's like fake it till you make it. And it was saying that bacteria, yes, they do evolve very, very, very quickly. I mean, extremely quickly. But some of that evolution is actually aided by the fact that they're sharing resources. They're sharing genetics, which is actually quite exciting. But they have this, if you're a bacterium and you have this F factor, which is this genetic content, which seems to do its own thing, it seems to be different genetics from the other genetics that you have to build your cell, then you can create what's called a pilus, which is this bridge that sends information from you to your neighbor. You just go up to this neighbor who doesn't have this F factor, and then you do have the F factor, and so that means you can plant this information into the other cell. And this is what it looks like in when you look at a microscope picture. It's just creepy. I mean, look at this thing here. It's like this one organism just minding its own business, and then somebody, it's like your neighbor. Imagine your neighbor like kind of builds a tunnel from his or her house to your house and starts like pushing all this like old junk from their basement into your dorm room. You might think, wait a minute, I don't want all this stuff. I don't need this. But anyway, this all happens when you have this F factor, the fertility factor, and this line that's built here is called the pilus, or the sex pilus, which is basically how you move the genetic content around. Okay, so anyway, it's just a bridge. This is actually quite a piece of research in itself, though, this F factor. Not every cell has it, but those cells that do have it can build these bridges. And if you're a cell that does not have it, then you're basically not going to be able to build a bridge. But it seems that having this F factor, or this fertility factor, which is denoted in our book by F plus or F minus. F plus means you have it, you have the factor, and F minus means that there is no factor, that you do not have the factor. But if you have this, then it allows you to actually move some of your content around. And so when they were trying to, in some of the research papers that you'll see, when they're looking to see how horizontal gene transfers have happened, they start by looking for, well, which are the organisms that have this F factor? Who could have sent the information from one side to the other? When I say information, I'm saying who could have sent the genetic content from one cell to another. Here's another picture of this of the cell that has this F factor. The donor builds the pilus and sends everything across. And here's this electron microscope picture. You can see how these two, another cell is just minding its own business when, hey, I've got my genetic content for you. I'm like, oh, great, thanks. It makes you wonder, though. I mean, how did the evolution, what were the evolutionary pressures that formed this kind of system here? Was it strictly to survive different kinds of toxins that may have existed in the environment, or was there something else that created this whole mechanism that moves bacterial DNA from one organism to another based upon this F factor? These are just a bunch of pictures of this whole thing. I know that you're probably getting tired of looking at these things, but I think that this is just an amazing, it's just an amazing study when you think about how these bacteria, they're not just cells anymore that are just sitting in a tank, but rather they are smart and they're finding ways of overcoming any hardships, and they're sharing information. It's kind of like there are many, I guess, like maybe like it's like GitHub, for instance. In computer science, for instance, we have GitHub, which is how we share code with each other. So, for instance, I could write something, and then anyone here could then use that code, or you could write something, and I could use that code. But we would all leave our code for each other on GitHub or any of those other code development websites. And here it's the same kind of idea where the cells are sharing information. So anyway, this is just more of this, but we're talking about the relaxosome. I'll just read this. A relaxosome, a complex of proteins that facilitate this transfer of information from one cell to another. And so it's not just like a random fluke here, but this is like a long evolutionary time to develop a protein like the relaxosome, which actually makes it possible to actually move genetic content from one organism to another, regardless of whether that's the same organism or the same type of organism or not. It could be from one type of bacteria to another type of bacteria. But the thing is, though, that it's had a lot of evolutionary time to actually create the proteins and I guess the method, if you will, to make all this stuff possible. But one thing I will say that's absolutely astounding is not necessarily, I mean, it's interesting to see that you can actually move, that horizontal gene transfer happens in this way. But it's more interesting to me to see that it doesn't matter the species of the bacteria, the code is the same. It's like they're all speaking the same language, and the DNA is the same. And in fact, if we had some kind of horizontal gene transfer from bacteria, which, I mean, I guess could happen, where the bacteria somehow plug in data or genetic content into our cells, then we would also be able to use the same kind of, the DNA would be the same. The DNA is the same language. And in fact, this is actually very true. In fact, if you're thinking about diabetes, for instance. Diabetes, for diabetics, they have to take insulin. And back in the 60s and 70s, and I think into the early 80s, the insulin that diabetics would have to take had to actually have been isolated from animals. It had to be taken from animals. Like, you'd actually have to sacrifice pigs. That's a nice way of saying you'd kill the pig. But you'd sacrifice a pig, and you'd find the pancreas, and inside the pancreas you'd find the insulin. So it's a lot of pigs that had to die in order to make the insulin that's necessary for people. But then things changed a little bit, where now we have synthetic insulin, which means that we found the actual genes that create the insulin proteins. Everything involved in making insulin. We found those genes, and those genes have been taken out of a human and put into an E. coli, which seems to be this bacteria that you find everywhere. It's in your stomach and everything. I think they've done this with other bacteria too. But the E. coli now start producing the stuff that the genes dictate for them to create, i.e. they start making insulin. And so the E. coli have no idea what's going on. They're just living their lives, sweating insulin, and then you have these giant vats, like tanks, and they're just the insulin is being created into, and then you just scrape the insulin seemingly off the top. And there's a lot more than that. But seemingly, though, you're using bacteria to create a human protein, which is just extraordinary, because you think, how can it be that bacteria is able to even follow human proteins? I mean, we're so special, right? No bacteria can follow what I'm doing, right? Mommy said I was special. But actually, though, the bacteria and us, we all speak the same genetic language, which is quite exciting. And so that's how things like this, how horizontal gene transfer work, where you have the ability just to send code from one organism to another. This is just the summary of everything I've just been talking about. So here's the integration of DNA into a chromosome. Once that DNA is sent across from one cell to another, then of course it still has to be joined into the plasmid, that is the genetic content of the organism or of the bacteria. But that seems to happen. I mean, you have this DNA, this episome or a DNA fragment that can be kind of exist as a plasmid, or you can integrate this into the chromosome. But basically, you're saying that it's a chunk of new code that can just be spliced in. And once it's spliced in, then that organism starts doing whatever those genes dictate it to do. Just like how when you splice into E. coli, when you splice in the genes for making insulin, then E. coli starts making insulin. It has no idea what it's doing. It's just making insulin. And to an E. coli, that's perfectly normal. That's what I'm supposed to do. I'm making insulin. That's what my genes say. And so you can imagine that if those genes that are being spliced into some bacteria are giving it some kind of an advantage, maybe in avoiding being killed by some therapeutic remedy that we might use, like some kind of penicillin or something, then those genes will also provide the organism with this advantage. In the case of splicing in the insulin, that's not necessarily going to make the genes or the organism any faster or lighter, but it does help them to. I mean, it doesn't kill them, and so they survive. But if you have a gene that actually says, that teaches the organism, in some way, how to avoid being found by the body's immune system, then you get an organism which is stronger and faster. And one thing I should say, though, is that in the plasmid plasmid is your genetic ring, if you will, that exists inside a bacterium. It's where the genetic content is. It's the DNA, if you will, of the organism. And so in these plasmids of bacteria, there are lots and lots of genes. And so seemingly, if you cut this if you take this giant ring here, you just cut it seemingly anywhere, you're going to be getting a lot of genes. And those genes, if they get pushed into another organism, those genes, whatever they may be, will be transferred to the other organism. Now, in many cases, those genes may be the same. Maybe they're genes that create cells or whatever. They just they do basic housekeeping stuff. But every now and then, one of those genes could be this resistance gene. Did you know that if you grow your proteins like this, then you can avoid penicillin, or you won't be killed by ampicillin, or whatever it may be? And so that knowledge, I'll call it knowledge, but that genetic knowledge can be transferred across just randomly. And so, and that's how these plasmids, that's how this transfer can work. And so you get this genetic resistance or this antibiotic resistance. And that all happens because the plasmids had some code in there. So there's a bunch of different ways that this could happen. Remember, we have insertion, the sequences, like entire sequences of this, for instance, of this plasmid, entire sequences are just inserted. And that means that if they are inserted correctly, then the organism will survive. And then if those insertions provide some kind of help or advantage, then they will live on. We also have this pathogeneticity islands. These are like special areas where you might find where chunks have been sent in, or where they've landed inside. In an island, when you say an island here in DNA, when you say that you found an island, it means that you found that the DNA in this specific region is very different from the surrounding DNA. It's kind of like an island in the middle of the ocean where it's all water, and then suddenly you find this place. So you find like a kind of an area where the code is completely different, and that's again how you can see that this horizontal gene transfer has actually happened, because you're getting these islands that just kind of show up. But also, though, most commonly you see toxin resistance. That's like you see genes showing up in organisms that shouldn't be there. These genes came from, and I suppose you did some analysis, you find out that a gene came from organism 1, and suddenly it's found in organism 2, and you're thinking, how could it have got there? There's no reason for this exact copy to be there, but yet it does come over there because, or did get over there because of this. It was just integrated, that gene was integrated into the chromosome because it was on this plasmid that was somehow chopped up and sent over. Anyway, viruses move the information around, and then you have some other ways of, you have some other transfers where the DNA is just kind of floating around and it gets absorbed. But remember, though, that the genetic code that you see here did not come from a common ancestry. So let's talk about, like, for instance, this non-common ancestry. Let's talk about some horizontal gene transfers that can actually hurt us as people, and that is that, supposing that you have some kind of I guess meat, for instance, or milk, for instance, where it comes from a cow, and the cow has been given all these antibiotics, like again and again and again, all these antibiotics, and somehow horizontal gene transfer happens inside those. Inside the gut of the cow, and so therefore the antibiotics no longer work. So basically, the cow has generated this kind of environment where the culture is now resistant to antibiotics. Now, if you eat something from that cow, or you drink the milk, or some of that bacteria gets inside you, then you also have resistant bacteria inside you. It's like you've never taken antibiotics. And suddenly you get something from the cow, like the milk, or maybe it's in the meat, or something else, you have some animal product, but it's inside you, and that means that now you have resistant bacteria introduced into your system, and that means now, for you, those antibiotics will not work. It's like it doesn't work anymore. You can't use penicillin. Another thing is, like, in some places, for instance, pigs are given a lot of antibiotics, and they do that because they want, if you have antibiotics, then it means that your body is spending less time fighting off infection and more time growing. That's why, for instance, in some, when you look at some generations, like, for instance, my brother is younger than, obviously younger than my parents, but he's just a giant. He's just he's like a giant. And a lot of his buddies, I mean, he's in his 20s, a lot of his buddies are also giants. I mean, they're much taller than their parents, and their parents are much taller than their parents. So when I think of my brother, and when he's hanging out with his buddies, and then I see his buddies' grandparents also, in the picture someplace, the grandparents are not as tall as all these basketball players. I mean, they're just huge people. I'm not as tall as he is, but still. But the thing is, though, that when our generation, when we were growing up, we were given antibiotics so that we would spend less time fighting off infection and more time growing. But in some places, if you're always fighting off infection, that means that your body has less energy to actually grow tall. And so that's why people are getting tall today, because of antibiotics. Now, they do the same thing with pigs. They want bigger and bigger pigs, which is fraught with ethical problems, I would say. But still, you have this, you're giving these antibiotics to pigs, and then the pigs will also grow tall because their bodies are not fighting off infections. They've got the antibiotics. They're just growing big. But the thing is, though, that the cultures that exist inside the pig's guts, like the bacteria, they become resistant to these antibiotics, and then you get something of some product from that pig inside of you, like maybe you eat the meat, or you have something else, who knows? And it doesn't take much of this bacteria, but just enough to make a colony inside you, and then before you know it, you are resistant to bacteria, and that means you are resistant to ampicillin or penicillin or whatever these antibiotics are. And so the next time you get sick, you take some kind of medicine that the doctor gives you, and nothing happens. You're still sick. And they're all scratching their heads thinking, it's funny, actually, because normally you should be getting over strep throat. I don't understand why you still have strep throat after 3 weeks of taking this penicillin. This is actually quite interesting. These are some interesting videos you can watch. I'm not going to watch them right now. I think this one over here, the first video is a little bit longer. It's more of a kind of an 8-minute video, I think it is. But actually, it's quite interesting. It's from the University of Exeter, which is in England. And it's kind of funny, but it's very knowledgeable. Next one is about this antibiotic resistance, which is more of a cartoon, which discusses this kind of scenario. So why are we talking about horizontal gene transfer? I've just spent all this time talking about this. I'm thinking, why are we spending all this time talking about this? Like, why do we care? The reason why we care about this is because it makes one sequence different from another sequence. And so it makes our analysis more complicated. So we have to be able to kind of, in order to figure out like how things are related to each other, or to determine like issues of relatedness, we have to compare the genetic sequences and be able to find out how one thing is different from another. Now, if you have a horizontal gene transfer, then you have to tell yourself, okay, fine, I know that I recognize that as horizontal gene transfer. I can ignore that. That's not telling me how related these guys are. It just tells me that the two organisms were at one time in the same dish together. But you have these, but we're really talking about like comparing sequences here, things that make things. And so we have this tool here called Clustal Omega, which is quite interesting, that checks for common sequences of genes. And maybe, for instance, those common sequences actually did come from horizontal gene transfer. And we've had this, and we've seen this. In fact, you've seen these slides before. I just put them in here just for continuity, where you can actually check to see that the two different sequences are the same. And we get these kinds of results too, where you can kind of say, okay, well, I'm comparing the proteins of these two sequences. I want to see how these proteins are different, but essentially it's the same thing as comparing the actual DNA sequences. And you can see that this middle area, where all the gaps are, that middle area where everything seems to line up between tests 1, 2, and 3, that could be your horizontal gene transfer island. That could be the stuff that could be a whole chunk of code, which was somehow recently, or between now and when they drifted apart, these organisms, but that could be some horizontal gene transfer evidence. But notice we're only like looking at, like, we're only comparing like 2 different or 3 different organisms. But what if I told you that we can now compare one sequence to every sequence known to man? Every single sequence. You can. It's called BLAST. And there's this giant database, which is it's a giant genetic database here that holds seemingly every piece of genetic content which has ever been discovered has to be entered into this giant database. And so what you can use BLAST for is to find out whether, has anyone ever found this same chunk of code that I have in my hands right now? Anywhere. Has anyone ever found this? And so what I wanted to do is I wanted to spend some time here to kind of show you how this works. It's actually a very exciting thing. But remember, though, this compares one sequence to a database of over 100 sequences. I'm comparing not just one sequence to another sequence, but I'm looking at my sequence compared to has ever been found in pig, in bat, in bacteria, in this, in that, in wherever. And even then, what type of pig, what kind of bat, what kind of fish, you'll find that there's many different ways that this gene can show up. It's almost mysterious if you didn't have these kinds of tools to show you how easy it can be. Find the best hits and find some optimal alignments. Let's just go out to this website and I'll just give you an idea about how to use it. In fact, I'll just go ahead and click on this. Oops, you know what? I don't want to do that just yet. I don't want to lose my slides. Get back in there. Okay, so I'm going to go here. So this is what the BLAST tool website looks like. What I'm going to do is there's actually some slides that I have in this thing that you can see where this is all coming from. But if you just go ahead and click on the Nucleotide Blast button, this is the You didn't see which one I was clicking on. I was clicking on just underneath where I am right now, Nucleotide Blast on the left-hand side. Nucleotide to nucleotide. That's your DNA. You can use a BLAST search for protein, which is, we'll talk about that when we hit protein. But this is actually gets very, very exciting. And in here, you can enter the accession number, which is the number of of the particular organism that you're looking at. So let me just go ahead and try something here. You'll notice that along the top of the page here, there's this COVID-19 is an emerging, rapidly evolving situation. If I go ahead and click on this data up here, I'm going to find the COVID. I'll just click that. I'm going to find a COVID sequence, and we're going to run BLAST on the sequence to see whether there's any other particular organisms which are to what we have as COVID today. So you go ahead and click on this area right here called nucleotide records. And here's a list of all the known sequences which exist as SARS or variants of COVID. In fact, the very, very first one here, if you click on this, you'll see this piece come out. And so my record, I'll just go ahead and copy this into memory, but my record number is NC_045512. If I go ahead and click on that, I get the record that comes up for the virus which we know to be the COVID-19 virus, which was discovered in Wuhan. So this is the record, and we've seen this before, and we'll talk about these records, I think, next week. Is that what it is? But you'll notice that over here on the side, where it says analyze this sequence, oops, right here it says, it says run BLAST. Can everybody see that? Yeah. You can run BLAST, and so I'm going to go ahead and click on this BLAST here, and that takes us back to the same website where we just were, just earlier. And so what we're going to do now is I'm going to go ahead and just leave all these settings the way they are, but what I'm going to do, though, the accession number is this number, that like this primary key, if you will, that exists for each sequence. Each sequence has a kind of an alphanumeric key, which allows that sequence to be kind of stored in a database. You leave that the way it is, and that's what your accession number is, but you leave that piece of information right here. And I'm going to go ahead and leave the nucleotide collection, the NRNT. There's a bunch of different ways. We'll talk about some of these, but these are different ways of actually comparing sequences here, but nucleotide is the more general one that we're using for this. And then over here, I'm going to leave this option of highly similar sequences, Mega BLAST. I'm going to go ahead and just see what happens here, and I'll probably have to keep talking while this is going on because this is, I ran this last night, and it does take some time. And so I'm going to leave this going. And in fact, you can run this too, and if it finishes sooner for you than it does for me, then that's just not fair. That's just not fair. But I'll tell you though, it does take some time though, and you'll notice that there are times during the day when the time is that it takes to complete some of these analyses is different, and that's because you're sharing these servers with thousands of other people at the same time. It has finished. Okay, so this finished in about I don't know, 20 seconds. Not bad. Last night it took about a minute. So what we get down here though is, well, let's just go through this again. So we have the job title. So this is like talking about what is it that we're looking at here? What is this analysis? This analysis is, this is your Wuhan genome. That's the genome of the virus. This is some of the information about, we're actually creating a search here, the program that we use, the database, the query ID, the description, okay. But the actual data that we're looking at is here, where it's saying, okay, these represent all the other viruses that somebody has picked up, like researchers have picked up from patients, and it doesn't just stop here, it probably goes on even further. But these are other variants of the COVID virus, other variants that exist out there that are known to science. They know that this sequence exists in other organisms. Maybe it was this horizontal gene transfer that made them different, but they're not the same sequence. They're very similar, but they may have popped up in different areas, which makes them different. And so, for instance, you can see in the descriptions where I am right now, hopefully you can all see this. Right. I'm looking at the description on the left-hand side, but each one of these viruses has its own name. So the first one is the, where is that? We have the WIV04, then we have the Wuhan, then we have the SARS-CoV-2. They're different viruses. I mean, they may be related. So what you want to do is you want to look at the first thing I look at is something called the E-value. And the E value is, remember, is the smaller the value, the more similar the sequence. In other words, the sequence, this sequence I'm looking at under my job title, the one we just keyed in, or this sequence here, the query ID, remember this, the stuff here that I found? That's the stuff we actually picked up from the website, from the COVID variant that was discovered in Wuhan. So all of these sequences here are being measured against that one sequence to see how similar they are. So if we didn't know, for instance, that this virus was a COVID virus, we just run it through this blast checker here, and you look at the kinds of descriptions that come out that you see on the left-hand side, you'll see that these are all SARS, COVID, CoV-2. It suggests that the sequences are absolutely the same. So getting back to this E value that I see over here, this E value, the smaller the E value, the more similar these different organisms are to this organism that I'm looking at here. So small E values mean small differences. All of these, in fact, all of these scores you see here all tell us the same thing, and they all tell us how similar something is. We have this query coverage. It's basically saying how much of the code is the same, how much of the code is different. And if the code is different, is it the coding regions or is it the non-coding regions? What is it? Like, why is it? It's very, very minor. 99% of the code is different, which means that it's practically the same thing. But because this E value is so small, it's saying statistically this is very similar. It's almost exactly the same. But actually, though, it isn't exactly the same because you have 99% similarity. But you'd expect that because it's a different variant. So like 1% has changed in some way. Maybe that was horizontal gene transfer. Maybe that was just mutations, whatever it was. But you can see that, for instance, for all practical purposes, 100% is the same. This is your percent identity. And then you have, for instance, you have the length of the sequence you're looking at here. But most importantly, though, you have under this column here a session. You have the other, I guess, the alphanumeric keys that you can use to find the original organisms which are being compared in this list. So I'll just look at the first one here. If I go ahead and right-click on this, I'm going to open this in a new tab. I don't know if you can see what I'm doing. You probably can't. But I've just clicked on the very first one, this one over here, this first record there. Just click on. Can't really see this very well. Sorry about that. But synthetic construct, ORF1, ABS, spike. I'm not sure how to read this. But this is, according to my BLAST search, where I've compared my sequence with every other sequence on record, this sequence came up as being very similar. And so if you look at this at the DNA itself, it's about the same length and about the same consistency of COVID. And so it's very, very similar. And so the nice thing about this whole, getting back to this BLAST searching area, is that I didn't have to know that I didn't know that for instance this organism was the same as another organism, my COVID organism. I didn't know that they were the same, but yet BLAST says, here we are. I just found another sequence which is exactly the same as this. It's kind of like, for instance, you're working in a library. A book comes across your desk with no title, and you run it through BLAST. You take all the pages and run them through BLAST, and then BLAST says, oh, you know what? This is this is this book on the back shelf here. It's the same story. I've read this book that you gave me, and I've compared these pages to every other pages or all the other pages in the library, and I found that it's this book. So therefore, you don't need a title because I found out by comparing the pages that it's actually this book that you have. It's a tremendous tool, and you'll find that if you read through bioinformatics textbooks, there's a lot of talking about, and there's a lot of information that comes from BLAST, and even many papers use BLAST like all the time. You can look at, for instance, these other links that are here. I'll look at the graphics one first. Let me go ahead and just click on graphics. Is this going to give me any graphics? Maybe it doesn't work here, but sorry. Let's look at the distance tree results. Let's try that and see what we get. What is this going? Doesn't seem to work. Sometimes it works and sometimes it doesn't. I guess in this case it doesn't work so well. But what you should see there. Let me just go back to the slides here. Maybe that would be helpful. If you click on some of these things here, this is just a simple workthrough that I did. I just randomly typed in some AT like that, just to see where that shows up. And that it turns out that by me just randomly typing in these bases, I actually did find that it does show up in other organisms. It came through all these other organisms here. And so by clicking on some of the graphics and things, you get pictures that look like this. or this isn't a picture from graphics, but this is a picture nonetheless. But the graphics, if the graphics clicked, or the graphics button worked, then basically you would have a picture or a snapshot of all the organism genomes that have common regions to code that I've been looking at. And so, for instance, here it's kind of hard to make out what all this stuff is, but this green stuff that we see here, those are the AT regions which are spread out across all these different organisms. And you can see in the sequence, like basically in front of where we're looking at right now, you can see that there's a bunch of AT. It's just repeating. It seems like it's like the genetic code is just becoming binary. But you can get graphics, and so you can actually visualize in the sequence where are these chunks of common code. And if you know, for instance, that an organism has chunks of common code at like around 80,000 bases into the genome, then like where you can see around, like, actually it's not 80,000. I guess this would be more like 100,000. But 100,000, if you know that that's a trademark, then maybe that's the organism you're looking for. Maybe, for instance, there's too many organisms that have this AT, but there's one organism that you know this AT content actually exists at a specific point, and so that would be something you'd use to locate this stuff. You also have trees, which exist, and if trees were working, I think that they do work, but it's just that they don't work with the data that we use, and I don't want to spend a whole bunch of extra time here to do another one of these searches. But I do invite you to do the same thing. The tree will look like this, where you click on the tree button, and that's your phylogenetic tree that you'll see. Where is it? Somewhere, the button that we were just looking at. But the phylogenetic tree, in fact, it'll be, not that one, hang on. Is it this one? No, it's not that. It'll be here, the distance tree results, if you can see where I am. It'll be there, just underneath where it is. But if you click on that, then your results are going to look like this, where it actually draws up a family tree, and it says it finds all the organisms, or all all the organisms for which the sequence was found, and it will show you a tree of relation. And so in this case, you're seeing that the NZCP010184.1 E. coli strain M3 plasmid complete genome, this is also, that chunk of code was found in the enterobacteria. Well, the thing is, though, that I wasn't surprised to see that E. coli, the same bacteria that lives in your gut, I wasn't surprised to see that E. coli had that chunk of code, because it seems that every time you do any random code or any code that you might find in the A bacteria, you're probably going to find an E. coli too, because E. coli is like the biggest. It's the biggest collector of all kinds of genetic content from seemingly all of its neighbors. It's like the guy who just like has to collect everything. And so the E. coli, I wasn't surprised to see that E. coli had this particular sequence of DNA, even though it came from other organisms too. So anyway, here's a BLAST demo. Maybe we have 2 minutes here. Let's see whether we can get this thing to work here. This one I know it doesn't take too much time. So let's just all try this right now in class. Let's just run our own BLAST. So go to the BLAST website. What I do if I'm looking for BLAST, I just type in an NCBI. I'll just show you. I'll go here. Type in NCBI, do a search there, and then you find this website here called the NCBI, National Center for Biotechnology Information. And on your website, you'll find BLAST is like right here on the right-hand side. Hope you can find that. You go here, and then you find this web BLAST. Click on your nucleotide, and you're in. So I'm going to type in this sequence, and the sequence I'm typing in is KF88. Hang on. KF88. What's the other stuff here? 19701. 1970.1. So what is this? This is the call sequence from, or the identifier of a specific sequence, which I think is called the CERT protein. Leave everything the same. Leave everything. Don't change anything. But I'm looking at what it is here. It's a protein which I have come across in research. It's the CERT. Yes, that's it. It's CERT. So what this is this is actually an organism called the CERT1, which is basically sorry, not an organism, it's a gene that does a whole bunch of things that's actually very handy in many ways in your body. It's a regulatory type of gene. Anyhow, so let's just say that it's all working. I'm just assuming that everything's working. You'll see that there are many different organisms. You have different scientific names here. These are all the different types of organisms that you'll find in the scientific name part where I am right now. I don't know if you can see where I'm actually clicking on. It's so annoying I can't show you. But in the middle where it says scientific name, these are all the organisms from which that same sequence for cert1 has been found. And if you look through, you'll see that there's a lot of different types of species here. I see birds, I see types of bacteria, I see rabbits, and I see some other kinds of birds. I see a whole, a great variety of different organisms. But remember though, look at the E value, which is like right here. And this is saying 0.0. This is called the first one on the list here. This is the Cyprinus Carpio cert1 mRNA, partial CDS, partial coding region. This is a low E value, and that's saying that it's, or 0.0, which means that it's practically, it's the same thing. It's like and this says 100% of the sequence is identical. So 100% of this sequence here at the very top of the list is actually the same thing as this organism that I was, or this genetic content I was looking at here. Anyway, we're kind of running out of time here, and you can go ahead and click around. I mean, go ahead and you can click on the distance, the tree, and you'll find that the tree actually does get made. So you can see kind of like how close, who else, and basically you're asking the question like, how closely related are these organisms that have this the same gene? And if they have this gene, what's the gene look like in them? Is it the same gene, or is it similar? So really you're saying, who has this gene that I've just found? And so by doing this, you'll see that cert1 actually does seem to be a very common piece of, I guess, biological machinery that you'll find in many different organisms, which is interesting. Okay, that's it. When we come back next time, we're going to be doing some more work with BLAST. Please do spend some time looking through this. I think you might find that there's a bunch of really amazing stuff to be found. The rest of the slides are really just kind of talking about how you can use BLAST for other types of analyses, and then there's another kind of a tutorial at the end here that you can look at if you want more information on BLAST. That is all. I will see you all on Friday.